This video demonstrates the proper technique for extracting peripheral blood mononuclear cell pellets from cell preparation tubes, or CPTs. In this video, we will be processing 2 CPTs with 4 milliliter draw capacity from the same participant. CPTs should remain upright and be processed within 2 hours of receipt. Place tubes containing whole blood into centrifuge rotors fitted with the adapters for 13 millimeter diameter tubes. Be sure to screw the aerosol lid onto the rotor tightly in the unlikely event of tube breakage. Balance the tubes appropriately within the centrifuge. Centrifuge the tubes at 1700 G for 20 minutes with the centrifuge brake off. After the centrifuge stops, immediately remove the tubes and place in a rack. There should be 5 visible layers. Starting from the bottom, the first layer is red blood cells and granulocytes. The second layer is the clear fecal material. The third layer is the polyester gel. The fourth layer is the peripheral blood mononuclear cell, or PBMC layer, which is usually thin and cloudy. The last or topmost layer is the plasma, usually pale yellow. If the red blood cells have not fully entered the lower layer of the tube, you can repeat this centrifuge step a second time. Once the tubes are in a rack, remove the plug at the top of the tube and use a Pasteur or serological pipette to discard approximately half of the plasma into liquid biohazard waste. In this example, we dispose of the plasma into a bleach solution, dip the pipette tip in bleach to sterilize it, and then discard the pipette. Next, prepare and label 1 new small storage tube for each CPT sample. In this example, we are using a microcentrifuge tube. Using a pipette, carefully remove the 1.5 milliliter of plasma containing the PBMC layer and transfer it into the storage tube. Capture as much of the PBMC layer as possible and try to avoid pipette contact with the polyester gel layer. Discard the CPT tube into a glass or sharp biohazard waste container, discard the pipette or tip, and repeat the transfer process for any additional CPTs. Place the storage tubes into a microcentrifuge, placing them opposite one another for balance. Be sure to securely place the aerosol cover over the centrifuge bucket in case of tube leakage. Centrifuge the tubes for 2 minutes at 12,000 RPM, or the equivalent of 300 RCF on your laboratory's centrifuge. Remove the tubes from the centrifuge. Note that you can see the pellets within the Eppendorf tube. Open the storage tube and pipette out or decant out most of the remaining plasma. Be careful to leave a few microliters of plasma